Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjun classes. Dear student, in previous class we have started with the introduction of this very important chapter, that is the principles and inheritance. That is the principles of inheritance and variation. Now, dear students, in previous lecture we have already talked about all the terminologies which are going to be used in this chapter and what type of question can be asked from that particular terminology. But we know that in the genetics, the Mendel's experiment, Mendel's study is one of the important event, one of the important base for the whole genetics which is developed today. So the time period or era before the Mendel is known as pre-Mendelism, and after the Mendel, it is known as post-Mendelism. Okay, so several important things has happened during the time period of Mendel, and the full name of Mendel is Gregor John Mendel. It is Gregor, that is Gregor John Mendel. And this Gregor John Mendel, born in July 22nd July 1822. Okay, dear students, name is Gregor John Mendel. Now, if we talk about Mendel, Gregor John Mendel was born in 1822 and died in 1844. First, we are going to talk about some of the history of Mendel, and he was the father of genetics. Now, today also he is known as father of genetics because majority study is done by him. He was the first to demonstrate the mechanism of transmission of character from one generation to the next generation. He was the first who showed that particular character transmission can occur from one generation to the another generation. Okay, and because of this, he discovered the basic principles of heredity through the certain experiment in the pea plant. He worked on a vision pea plant. Okay, so this is the thing. And later on, after working on a pea plant, he published all the result what he discovered by his studies under the title Experiments on the Pea Hybrid. Okay, experiment on the pea hybrid or a pea plant. If we talk about Mendel's experiment, then dear students, for the first time, Mendel conducted hybridization experiment on garden figs for 7 years, 1856 to 1863. Now, if we talk about his study from 1856 to 1863, that is 7 years, he had worked experiments on the pea plant, and he proposed some of the law of inheritance in organism. Before 1856, he was in University of Vienna. Okay, and then later on he worked on this particular plant. Now we remember one of the important points that he was here. How many character? 7 character is written. Means if 7 character means total 14 varieties. He worked on a 14 varieties, or we can say true breeding varieties. Okay, on this he worked. He proposed the laws of inheritance in living organism by this experiments, and what Mendel did, which is unique to him, and Mendel first time applied statistical analysis and mathematical logic to the problems in biology for the first time. And due to the application of this mathematics and statistics on the biology, his work was unique. Now you can see this is the Mendel working on the pea plant. And in pea plant, you can see there is a hole. This is the pod. This is the pod of pea. And in pod, within pod, you can see this is the seed. This is the flower. So certain characters related to this was taken by Mendel. And Mendel worked with the genotype and phenotype. What Mendel investigated, he investigated that several characters in garden pea plant exist, and he also noticed that many characters are manifested by two opposite traits. Means, if we talk about any character, it has a two trait, two variant forms. Now the question is which variant form that need to be studied. So such that he given an example that tall and dwarf plants are the variants of the height. Yellow and green seed color is also a variant or traits of the character color of seed. Okay, so dear student, here if we talk about character, these are the traits. If we talk about character, then for this character is a height of the plant, whereas for this the character is a what? Seed color. So same way, total 7 character was taken, but 14 trait or 14 two breeding varieties have been studied. So dear student, you have to remember this data of 7 and 14 character, 7 character and 14 trait, because in need 2 times they have asked question from this simple part. Later on, a cross between tall and dwarf plant was carried out. You can see this is the plants, dwarf and long, tall. So tall plant was taken, dwarf plant was taken, and cross between yellow and green seeds also carried out. Mendel working with the pea plant in his garden. Mendel was working with a pea plant in his garden. And in pea flower, you know that in class 11th we have studied that this is the pea plant have a vexillary estivation. Pea plant have vexillary estivation. And you know that estivation is a arrangement of sepals and petals with the members of their own wall. And in this, you can see case of maxillary estivation in the corolla of pea. And you can see these are the stamens, and this is the particularly a petal. A smallest petal keel is there. So in maxillary estivation, what you will find? One is the largest petal you will see, two small petals you will see, and two smallest petal you will see. This is known as vexillum or standard. This is known as wings or LA. This is kill or carina. Like this, you will see the auxiliary estivation. And here he used the particular stamen and carpel for this experiment. So if we talk about character and their contrasting trait, then first character is stem height, which having a tall and dwarf character. Then flower color, which can be violet and white. Flower position can be at the apex, terminal, or axial. Pod shape can be inflated or constricted. Okay, so it can be swollen or constricted. Inflated means swollen. Pod color can be green or yellow. Seed shape can be round or wrinkled, and seed color can be yellow or green. So this many character was particularly taken and studied. You can see here total 7 characters are given and 14 varieties are here. But dear student, if we talk, then this all characters are located on specific chromosome number. They are located on specific chromosome number. The genes for this particular character are located on specific chromosome number. So that we have to write. First is plant height, and plant height genes that is dominant for tall and recessive for the dark trait. For this, the genes are located on the fourth chromosome. Which chromosome? Fourth chromosome. Same way, flower color, if we say violet or white, that is the gene of it is located on the first chromosome. Then flower position, if we take, they can be axial or terminal. It is on the fourth no, chromosome number. Pod shape, if we take, it is also inflated or constricted. The genes are present on the fourth chromosome. Then pod color, if we talk, the genes are present on the fifth chromosome. Seed shape, if we take, that can be round or wrinkled, genes are located on the seventh chromosome. And the seed color, yellow or green, that genes are located on the first chromosome. So, in case of this, particularly 7 characters are there, but these 7 characters are present on only 4 chromosomes. So, Blixt was the scientist who given that, or who explained that the genes of 7 characters are present on 4 chromosomes. That is first, fourth, fifth, and seventh. These are the 4 chromosomes on which the genes of all 7 characters are located. The one of the important character which was used by Mendel in his experiment, that is a stem height. And if you talk about stem height, you can see this is a tall plant which dominant trait is a tribar plant, that is a recessive trait. Other color like flower color we have taken just, that is a violet color and the white flower. Okay. Then flower position, you can see this is the axial inflorescence, means the flower are present in the axial, in the branches, axial of branches. See here, what is axial? See, suppose this is the terminal inflorescence, so means the flower is present at the tip of the branch, and here flower is present in axial of the branch. Okay, this angle is known as axial. This is leaf, and this is the growing part. So this angle is known as axial. And when the particularly flower originate in this axial, okay, then it is known as axial flower, okay, or axial inflorescence. Then pod shape, you can see pod shape is inflated, means swollen and constricted, you can see. Then pod color can be green pods or yellow pods. Green pod is dominant trait. Same way seed shape. Seed shape is a round seeds. Round seed is a dominant trait. Seed color, that is a particularly yellow seed or green seed. Yellow seed is a dominant trait. Okay, so this is the thing. Now, on this many character Mendel work, but Mendel started all experiment with the true breeding varieties. Mendel started with the true breeding varieties of this particular plant. Now, question is, what is true breeding variety? True breeding line is one that has undergone continuous self-pollination, show the stable trait inheritance and expression of several generations. Means, if we carry out selfing of particular plant again and again, again and again, again and again, then the homozygosity get increases and the heterozygosity get decreasing. Homozygosity is decreasing, means the character is getting stable. And if it is in homozygous condition, the chances of expression of it for the several generation are increases, and that is passing to the next generation through the gametes. So Mendel conducted artificial pollination or cross pollination experiment using several true breeding lines. Means all the true breeding lines was taken as a parent. A true breeding line is one that has undergone continuous self-pollination, shows stable trait inheritance and expression of several generations. Mendel selected 14 true breeding pea plant varieties as pairs, means 14 true breeding pea plant varieties as a pairs which were similar except for one character with contrasting trait. Okay, means if all are in particularly true breeding, all are particularly same. but one is differing. So means we are focusing on, suppose out of seven characters, six characters are similar in both the plant, but only one character is differing. If we make a cross between these two plants and carry out a hybridization, then in offspring we will see the effect or a change in inheritance or a particular type of inheritance from the parent to the offspring in a such a way for the particular character. Now, the question first which arises in our mind that why Mendel experimented for 7 years on the pea plant only. Okay. So advantage of selecting pea plant by Mendel for hybridized experiment are first, it is an annual plant that has a well-defined characteristic. It is an annual plant. Okay. Annual plant means we can say its life cycle complete within 1 year. Means within 1 year it is going to flower and get die. Okay. It can be grown and crossed easily. Easily it can be crossed. It is as it is a bisexual flower containing both female and male plants. Means it has a particularly both male and female sex organ, that is female and male plants, stamen and carpel. It can be self-fertilized conveniently, easily can be self-fertilized and a short life cycle and produce a large number of offspring. So due to this many characteristic, their short lifespan, many offspring, which help them to understand the inheritance pattern easily, crossing is easily, bisexual flowers are there, so self-fertilization easily. So all due to all of these many characters, the garden was selected by Mendel. But it is not that genetics started with the Mendel experiment only. Mendel also worked on another plant. Okay, he worked on the pea plant, but it is known that by suggestion of Carl Nageli, Mendel Mendel experimented on hawkweed also. Hawkweed or Heratium. Also, it is scientifically known as Heratium. But it doesn't give same result as pea plant. But it doesn't really give result similar to the pea plant. Because of this, they mainly focused on the pea plant. Okay. And performed sexual hybridization. Now, yes, after particularly working on pea plant, he was got a result. But by doing this experiment, he gone through the several failure and success. And the success of Mendel's work occurred that is due to some regions. And the regions are a selection of P for its experimental plant. And P has some different characteristics. P plant produces large number of offspring, so sampling size is large, which gives credibility of this data. Means the large number of offspring are going to be produced, large number of parents are going to be hybridized. So the chances of inheritance of character occur in many plants, then that is a correct credibility to the data. He started hybridization experiment considering single character at a time. Hybridization experiment means, as I told you, six characters are stable. Only one character has a trait, contrasting trait. Otherwise, six traits are similar in both the plant. And he performed a hybridization experiment by a considering single character at a time. Statistical analysis and mathematical logic were applied, which is given a unique feature. Okay, so these are the some of the particularly, we can say, reasons for the success of Mendel's work. So this entire work on a quantitative analysis and qualitative character. So because of this, Mendel got success. What are the pea plant characteristics that we have discussed? Why did Mendel select only pea plant for his work? It was fast-growing annual plant with a large number of contrasting features, which we have seen. It can be easily self-pollinated, that also we have discussed. Produce a large number of fertile offspring in very short time. Okay, so these are the characteristics. Okay, so this is the thing was done by Mendel. But it is known that after the publication of his experiment, the Mendel's work remained unnoticed for the longer period of time. Okay, due to this many features, also they had this many features, but still the Mendel remained hidden for a long time. And later on, the work of Mendel rediscovered by different scientists. So it is known that in 1900, Mendel's work was rediscovered by three scientists. That is Carl Correns, Hugo de Ries, and Teschenmark. In this, Carl Correns, if we talk, he made laws from the Mendel's conclusion. He made law from Mendel conclusion. And Hugo de Ries published this work in 1901 in journal named Flora. Okay, so due to this contribution, the whole work was rediscovered in 1900. So first we have to understand what are the regions that Mendel's work will remain unnoticed for the very long period. Okay, so that question we are going to answer. So the question is why Mendel's work remain unnoticed or hidden for long time. And if we answer this question, then we come to know about some of the points that first thing is that Mendel's work wa- was very much ahead of time. Was very much ahead of time. So many people was unable to understand this properly. Second thing, he used mathematical logic and statistics. And statistics. And this use of mathematical logic and statistics also made the work different and not easily understandable for the others. The journal, next point is the journal in which work was published had limited circulation. Means it is circulated in local area only. And because of this, also the majority people does not know about his work. Okay. Then next thing is he was unable to provide any physical proof of factor. Any physical proof of factor means gene. Means where these genes are located, he was not able to answer this question. So because of this, also majority people was not sure about his work. Then he himself was not also not sure about his work. One of the important was what? Low confidence. He was himself not sure about his work. Means he was in low confidence. And at that time, the popularity of Charles Darwin was an indirect reason. Was an indirect reason. Okay, so due to these things, the Mendel work was remained unnoticed for many years. Okay, so this is the thing. Okay. And these are the reasons due to which the Mendel work remained unnoticed. Now, after this, we are going to talk about the technique of Mendel, how he worked with this pea plant and carried out the experiment, how he performed an experiment that we have to understand. So next we are going to talk about technique of experiments and what experiments are done that we will understand after this part. He said that in this three steps, it occurs in three steps. It involves three-step process. In this first step, if we talk, first step is selection of pure parent. Selection of pure parent. Pure means homozygous parent. Means either if we take a character height, then either it can be capital T or small t small t. So that parent need to be selected. And this pure or homozygous parent are produced by continuous selfing, okay, and selection of pure plant. And by this step, that is continuous selfing, he developed he developed 14 true breeding 14 true breeding or pure varieties, or we can say pure line. Okay. Now, after the particular selection of pure parent, if we take a tall, then it is capital T, and if it is small or dwarf, then it is small t. Like this parent has taken. Only homozygous parent has taken. And next, a second step is performed. Second step involves the hybridization of pure parent. Hybridization of pure parents. And in hybridization of pure parent, if you see, then it is by emasculation and bagging technique. By emasculation and bagging technique of artificial hybridization. Now, in this technique, what actually is done, then if it is a bisexual flower, then if we take a bisexual plant and if we take a bisexual flower, then first, if we want to take a particularly desired pollen grain from the particular plant, and if we want to fertilize it with the egg of the another plant, then we have to remove the carpel of that another plant and remove the stamen of that particular plant, and only carpel need to be removed. So we have to use a particular parent plant to parent plant, and from one we are going to use a male gamete or pollen grain, from one we are going to use a carpel. From the plant which we all need to use a stem, and there is no necessity of carpel, and in the plant where we need to use egg, there is a carpel is important, no necessity of this step. So in a bisexual flower, first is anthers are removed, and first step is emasculation. Emasculation means removal of stamen before maturation. Removal of stamen or anther before maturation. Okay. And after removal of anther before maturation, we remove the anther. Then we bag it with the butter paper, the remaining carpel, the in the bud stage, we remove this stem and then the only carpel are removed. This carpel are bagged with the butter paper to prevent it from the contaminated with the another stamen and it allowed to get mature. After bagging, when those carpel get mature, then debagging is done, means bag is removed. After removal of bag, what we will do, the desired pollen grain of a particular plant which we have selected are dusted on the carpel stigma and that is known as dusting. After dusting, we need to allow them to fertilize with the desired male gamete or we need to allow them to develop pollen tube and then fertilization will occur. So we carry out rebagging. After rebagging, the fertilization we can occur, but before that, we particularly tag that particular plant that artificial hybridization is done. So this is the step in bisexual flower. In unisexual flower, if we are going to perform this artificial hybridization, then in unisexual flower, emasculation is not required. Okay, so this is the thing. So when we need to carry out a selfing of bisexual flower, then only we need to particularly do what? A removal of stem for emasculation. And after this, the third step which involved here. So after this hybridization of pure parent will occur, and after the hybridization of pure parent, as you can see in the second step, the hybrid F1 progeny will be obtained. So hybrid F1 progeny is obtained by hybridization of pure parent. Then third step we are going to perform here, that is selfing of F1 hybrid. So F2 generation is obtained. This is the steps which are taken by this one. So what if we get a F1 hybrid? From F1 hybrid, we are going to take their seeds, then we will grow that seeds, then fertilization will occur and F2 plants will be obtained. F2 plants, they have seeds that will grow and breeding will occur. So F2 plants will be obtained. And so on, things is going to happen. And when we carry out continuous selfing like this, we know that this is the crossing, hybridization actually, cross hybridization. But if we carry out continuous selfing as we have seen for the production of parent, then homozygosity get increases. But these are the three steps which are necessary for the carrying out experiment. Okay. So let's talk about some of the questions. Who is the father of genetics? If we talk about father of genetics, then he is nothing but Gregor John Mendel. Okay. Then the next question is Gregor John Mendel conducted his experiment on which plant? Then you know very well this is the nothing but pea plant. And this pea plant is also known as garden pea. And this garden pea is an edible pea. Then who proposed law of inheritance in living organism? Law of inheritance in living organism was obviously proposed by the Gregor John Mendel. What is the contrasting trait for semite? Just we have discussed many times. It is a tall and dwarf. Tall is a dominant trait and dwarf is a recessive trait. And the next question is, what is the advantage of selecting pea plant by Mendel for his experiment? The last one, short, because they need to self-pollinate, so cross-pollination cannot be there. Short life cycle needed, and bisexual flower are used, so short life cycle produces a large number of offspring that will be having a large sampling size that helps to, for the credit. This gives a credibility to this particular data. Okay, so short life cycle and produce large number of offspring. Okay, dear students, so this is the Mendel's work, of Mendelism, how Mendel experiment was carried out, what are the technique, what he had selected, how they worked, that we have discussed. Now, next onwards, from next lecture onwards, we are going to start the what experiment actually he did. Okay, so till that, thank you, dear students.